Welcome to our lesson on genetic engineering. The key questions for today are: what is genetic engineering? What is involved in the process of genetic engineering? Why is genetic engineering used? What are the risks of genetic engineering? What are the benefits of genetic engineering? The key words that you will need to know are genome, vector, gene, plasmid, resistant. Genetic engineering, also called genetic modification or GM, is a process which includes modifying the genome of an organism by introducing a gene from another organism. This is done to introduce a desired characteristic into the organism. This could be for a variety of reasons. Food crops can be genetically modified to increase their yield, to increase the amount of vitamins present, or even just improve the flavour. Plants and crops can also be genetically modified to make them frost-resistant or resistant to herbicides or pests. This can be very important for farmers who rely on crops for their livelihoods and also for the general population, as it can increase food security. Bacterial cells can also be genetically modified to produce enzymes or proteins that are used in medical treatments. An example of this is bacterial cells being used to produce human insulin, used in the treatment of diabetes. The process of genetic engineering includes selecting the characteristic that is desirable and that should be transferred from one organism to another. The gene responsible for that characteristic is then isolated and cut out of the organism's chromosome using an enzyme. This gene is then inserted into a vector. The vector then transfers the gene to the other organism. A vector can be a virus or a bacterial plasmid. These can be used to transfer the gene to the desired cells in the organism. This is usually done early in the organism's development, so that the cells replicate so that they all contain the gene for the desired characteristic. Task: choose from the following words to complete the sentence. And here's the answer. Genetically modified crops are produced by cutting the gene out of the chromosomes of one plant and inserting them into the cells of a crop. This way, desired characteristics can be transferred from one organism to another. An example of this is a gene for frost resistance that was discovered in fish. The gene was isolated and removed from a fish cell using enzymes and was transferred to tomato plants using a bacterial plasmid as a vector. The bacterium invaded the plant cells and transferred the gene. This meant the tomato plants then contained the gene for frost resistance and were able to keep producing edible fruit even in cold temperatures. Extension. Answer this AQA exam question. Glyphosate is a herbicide. Crop plants have been genetically modified to make them resistant to glyphosate. A. Why is it an advantage to make crops resistant to glyphosate? B. Figure 1 shows how scientists produce genetically modified GM crop plants. The scientists use a GM bacterium that can invade plant cells. 1. The ring of DNA shown in Figure 1 acts as a vector for the resistance gene. What is the scientific name for this ring of DNA? 2. At step 1 in Figure 1, the ring of DNA is cut open. How do scientists cut open the ring of DNA? 3. At step 5 in Figure 1, plant cells and GM bacteria are put on agar containing glyphosate. Explain why the scientists add glyphosate to the agar. Answers. It is an advantage to genetically modified crops to be resistant to herbicides, so that herbicides can be applied to the whole area and will kill only the weeds, not the crops. This means that the crops will have less competition for nutrients in the soil, light and water, so will grow more, which would increase the yield. B. 1. The ring of DNA in a bacterial cell is called a plasmid. 2. The plasmid is cut open using enzymes. 3. The gene would only be taken up by some of the cells, so by applying the herbicide, any cells that have not taken up the gene to make them resistant would be killed, so that only those with the resistant gene would be left and would grow into herbicide-resistant plants. There are many more advantages to genetically modifying crops. For example, in some developing countries, vitamin deficiencies are common, so scientists genetically modified rice plants to produce rice with higher vitamin contents. Genetic modification is a fast way to produce many organisms with desired or useful characteristics. It yields similar results to selective breeding, but has the advantage of transferring characteristics across species. In the future, genetic modification may be able to be used to overcome inherited disorders in humans. Although there are advantages to genetically modifying certain organisms to have desired characteristics, there are some ethical arguments against it. There is not a lot known about the long-term effects on human health of eating GM foods, and people worry that there may be harmful effects in the future. Also, some people may have an allergic reaction to substances that may be present in GM foods. There are also concerns that it is not right to alter the genes of living organisms and create new life forms. Task: types of fish can be genetically modified by inserting a gene found in jellyfish that causes them to change colour when certain pollutants are present. How could this be useful? Why might people disagree with the use of genetic modification? This could be useful as the fish could be used to indicate if water is safe or not, to indicate if pollution is present. Some people may disagree as they are worried about the genetically modified fish entering the food chain and causing allergic reactions or health issues in humans. Some people may also think it is wrong to alter the genes of the fish. Extension: read the following text taken from an AQA exam paper. Would you advise farmers to use GM maize plants? Explain your reasoning. Insects can be both useful and harmful to crop plants. Insects, such as bees, pollinate the flowers of some crop plants. Pollination is needed for successful sexual reproduction of crop plants. Some insects eat crops, and other insects eat the insects that eat crops. Corn borers are insects that eat maize plants. A toxin produced by the bacterium Bacillus thuringiensis kills insects. Scientists grow Bacillus thuringiensis in large containers. The toxin is collected from the containers and is sprayed over maize crops to kill corn borers. A company has developed genetically modified GM maize plants. GM maize plants contain a gene from Bacillus thuringiensis. This gene changes the GM maize plants so that they produce the toxin. Advantages of using GM maize plants include less effort for farmer or less likely to harm farmer. Pesticide always there or doesn't wash away and there is no need to spray the plants. Less insects to eat crop, maize or carry disease, so greater crop production or yield. Disadvantages of using GM maize plants include toxin kills other insects, so some crops don't get pollinated, sexually reproduce. Possible harm when eaten by humans or animals. Damage to food chains. Gene may spread to other species.